So before we start the details of DNA replication, I would like to answer the question from the previous video. In which cell, in which step, in which phase during the cell cycle DNA replication occur? Remember, in the cell cycle we have G1 phase, S phase, G2 phase, and then karyokinesis, mitosis, and then cytokinesis. So which one? So the answer is S phase. During S phase, DNA replication occurs because S stands for synthesis. So it's a phase that we make we make new DNA molecules. Okay, now we have this replication. It is summarized in these few steps here. So before DNA replication occurs, we have the double helix. Remember that they are anti-parallel, and then they form this double helix, and then they use hydrogen bond to connect the two strands of DNA together, and then we use an enzyme. This is an enzyme. This is one of the 3 enzymes that I want you to memorize for the DNA replication. Other than DNA polymerase that we talked about previously, this one is called DNA helicase. Again, ASC tells you that it is an enzyme. Helix, meaning that it has something to do with the helix. So what does it do? It unwinds and unzips the double-strand DNA, meaning that this enzyme unwinds the DNA double helix. That's why we call it helicase. Because this enzyme unwinds the DNA helix. And then after DNA is unwind, what will be the next step? Then the DNA polymerase, and then DNA polymerase will bind to the unwound DNA, and then you will bring the complementary DNA nucleotide and to fit into the original strand. So this is what happened. And then you keep moving along the original strand, and then as it moves along, you just keep grabbing the complementary nucleotide to form the new strand on the original strand. And then after DNA polymerase put all the complementary nucleotide to the original strand, or the template, and then there's a third enzyme that I want you to know is the DNA ligase. ASC again is an enzyme. DNA ligase, what does it do? It seals any breaks in the sugar phosphate backbone. So we will see this ligase in action later in the video. You'll have a better idea about it. And then the 2 double helix molecules are identical to each other and the original molecule. This is what I told you. It is very similar to mitosis in that the original molecule will become 2 daughter molecules. The 2 newly made daughter molecules, they are identical to each other, and then they are also identical to the original DNA molecule. So this is the overview. So you have the DNA helicase here to unwind the DNA molecule. And then the original strand is colored blue. The newly made daughter strand is colored as green. And there's one thing that I want you to memorize is that the newly made daughter strand, the green one, always made in the direction of 5' to 3'. So in here, you see that the mother strand here is starting with 3' here, and then 5' on the top. In other words, the daughter strand here, this end is the 5' end. So the newly synthesized DNA daughter strand is going from this end, 5', and then all the way up here. So you go to this one. We call this the leading strand. The leading strand because the DNA polymerase move in the same direction as the helicase. So they move in the same direction. And the leading strand is a continuous strand, so that when the DNA polymerase keep grabbing the complementary nucleotide, they can actually connect to the same strand without any break. So in this strand, DNA ligase is not required to work on this part. On the other hand, you see that this one, it has the 5' end on the mother strand here, on the template. So the 3' end is here. In other words, the newly made daughter strand, the 5' end is here. So what happens is that it will start somewhere and then replicate. And then when it reaches the end, what will happen? It will go all the way back to here, and then continue to make. And then what happens is that DNA helicase will continue to unwind while it is replicating. And then when it reaches the original starting point, then the DNA polymerase will detach and then go all the way back to where the DNA helicase just unwind. And you'll keep doing this and then keep replicating and then go to the higher point and then go down to replicate. So you can see that the newly made daughter strand will become a fragment. So you have the first fragment here, and then after the DNA polymerase finish making the daughter strand, you'll go back to where the newly unwind the DNA and then start to replicate. And there will be a break in here between the first piece of daughter strand to the second piece of the daughter strand. And then you'll go back to the newly unwind DNA and then bind there and then replicate it. So you have this kind of fragment. The fragment, we call this Okazaki fragment. And we require DNA ligase to seal up all this Okazaki fragment together. So this is what I meant. It's like mitosis. One original DNA molecule becomes two identical DNA molecules. So these molecules, this daughter molecule and this daughter molecule, they are identical to each other. And these two identical daughter molecules, they are also identical to the mother molecule. So this is what I just briefly talked about. Now you see the picture showing you this. So it's a helicase. It's keep on unwinding the double helix, unwinding the double helix. And then you have the leading strand here. The leading strand, because the green newly made DNA, it is all DNA made from 5' to 3'. And the 5' is here, the green part is here, this is 5' and this is the 3'. The DNA polymerase is moving in the same direction as the helicase. So while the DNA polymerase is replicating this leading strand, the helicase keep on unwinding, so they move in the same direction. But for this part, we call it the lagging strand. The newly made strand, we call it lagging strand because the 3' end is here. The 5' end is somewhere here. So you can see that the DNA polymerase, it has to replicate from here, and then it has to go back and bind again to replicate here, and go back to bind again to replicate here, and go back. Why does it have to go back all the time? Because the rate of this DNA polymerase to replicate totally depends on how much of the strand, the helicase unwind. So it may bind to here, and then replicate, and after it reaches this end, then it has to go higher, because by that time helicase already unwind the new pieces of DNA, and they can bind to the new pieces of DNA and then replicate, and then after this it go back and to replicate more. So it really depends on this, how much it unwind the strand, so that the DNA polymerase can keep on going back, go back to the so-called newly unwind DNA as a template to replicate the lacking strand. And as I told you before, these fragments, they are Okazaki fragment because it is discovered by a Japanese scientist. And then for this fragment, we require the DNA ligase to seal this fragment. That means to connect the fragment together. Okay, let's watch a video to help you to understand.
Cells like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce